So the name is immunosensor. So I think everybody know the word antibody, okay? Because yeah, during COVID, everybody tried to increase your immune system, trying to have a good antibody so that you can, um, you know, try to avoid getting infected. So one way is to get a good uh, antibody or immune system in your body. So first and foremost, like DNA, we have to know the basic structure of antibody before we move to antigen. Okay, so this is antibody. Okay, the 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 cartoon or the representation as well as the 3D structure. Okay, right. If you look at here, is actually chemistry, right? What you can see here, most probably quite similar to DNA, but DNA is a helix structure, right? But antibody is a Y shape. Okay, this is Y shape structure. Okay, so what is inside? What are the chemical functional group that is inside antibody? Okay, so during the in the Y shape structure, you can see there is amide bond, not amide bond. This is sulfur bond, and then you have carboxyl. Okay, so carboxyl group here. Okay, and at this side is amine. Okay, here. Okay, so the structure is very specific for antibody. It should have carboxyl group and it should have amine group here. Yeah, okay. So this is very important because we we are going to discuss antigen. So anyone know what is antigen? Antibody is quite clear. Everybody wish to increase your antibody, increase your immune to fight disease. But anyone know what antigen? What is antigen? Any idea or anything that comes to your mind? What is antigen? For those who
The
are not driving, yes?
the opposite of antibody,
The
Prof,
opposite, yes.
maybe.
Yeah, it's okay. I mean, questions. Okay, anyone has any idea on antigen?
Something in
Lily?
the body that's harmful, Prof?
Body that?
Something in the body that's harmful.
Okay, yeah, which is correct. Okay, so now, of course, you know, antibody is the one that you wish to have against, for example, COVID, dengue. Okay, okay, those are the one that is here. Okay, so we talk about antigen. Yes, it is true. It is the virus itself, the bacteria itself, the 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 toxic, the fungus, whatever that it is a pathogen, yeah, that trigger the infection, that cause the negative, the harmful effect. Okay, because antigen can be also in a form of what we call protein. Yeah, it's just a protein. It's not the whole bacteria. It's not the whole virus. It can just be um, a protein. So that triggers most probably negative impact. So anything, not to say anything, but those are what we call as antigen. And when we look at antigen and antibody interaction, they are like lock and key. So specific antigen will be interacting with specific antibody. Okay, so you see here where the amine, okay, let me, does it want to move? Okay, never mind. So this is the Y-shaped antibody. And as I shown before, we have carboxyl and we have this NH2. Okay, this is the general structure because of course there are some specific, what we call as binding sites. Okay, so this antigen, okay, whatever it is a virus, it is a protein, it is a bacteria, it is a fungus, toxin, whatsoever, will interact with this area. Okay, so it's like lock and key. So you can imagine lock and key, they are very specific interaction. Okay, so that's why when we are vaccinated, by right, our body will produce antibody that will bind to the antigen that the consider like a defense system, yeah, to our body, for our body. Okay, now if you look at this structure, okay, now the whole bacteria cell, the purple one, is considered the antigen. Okay, it's big, right? So it's not going to fit into a small area of antibody. Okay, that's why on top, or on the surface, not on top, on the surface of the bacteria cell, yeah, called, the whole thing is antigen, okay, can be called as antigen. But if just some part of the bacteria is separated from the bacteria, it is still called an antigen, okay? So what happened is, on the surface of the bacteria, there is what we call as epitopes. Okay, so you can see that the epitopes, there are many shapes. Remember that I have described to you the structure of sensor, where we have different shapes representing different analytes. Okay, same goes to the bacteria. You can see that on the surface of the bacteria, there are different types of epitopes. They are all antigen, means that they are the one causing the probably infection. But you can see that the antibody, okay, does not react with all the epitopes, because you can see you have cube-like epitopes. It's just for the purpose of discussion. And cone-like epitopes, probably you have round-like epitopes, but specific antibody will interact with specific epitopes. So in this case, for the purpose of discussion, this antibody responds to cone-like epitopes on the surface of electron. On the surface of the bacteria. So this is, you imagine a bacteria. You all, probably yeah, I do cough a lot, and sometimes I have to take antibiotic. Okay, so you imagine that probably there is infection in my throat. So there is some bacteria. So you imagine that bacteria has some epitopes on the surface of the bacteria, and the antibody is the one that produced by your body, will be responding to specific epitopes on the surface of the bacteria. Okay, so I hope you understand the concept of interaction between antibody, in short I put AB, and antigen. Okay, okay, all right. So you can see here this is the antigen, and this is the antibody interacting with the antigen. So this is the concept that we use for immunosensor. If it is DNA, I hope that you still remember that we are based on hybridization of the match, okay, DNA. But for immunosensor, we are focusing on the interaction of antibody and its specific antigen. Yeah, right. Now, this is another information on antibody. If you don't understand, please do stop me. There are what we call as polyclonal and monoclonal antibody. So the difference at this moment, I'm not going to be too detailed on the molecular part because I'm not a molecular scientist. Okay, when we talk about polyclonal, meaning that there is a possibility that the antibody can interact with more than one epitopes. So that's why you can see it has a lot of binding site. Means that the antibody probably can interact with more than one epitopes on the surface of the antigen. But monoclonal antibody, it is very specific. It really target a specific epitope on the surface of the antigen. So that is polyclonal and monoclonal antibody. Okay, of course, this one is more expensive, difficult to prepare, to produce, but since it is very specific, it is of course much more what do you call of use because of the specificity. Okay, so polyclonal is more of a bit general, because it reacts with many epitopes of the antigen. Okay. All right, so this is the one, this is the morphology of SARS-CoV-2, that the virus that cause COVID. The one that has attacked us, that has caused a lot of deaths throughout, globally. Okay, so this is the picture. This is how it looks like. I think people have looked at these pictures so many times, but what is the information from this morphological structure of COVID virus? So you can see here, this is the structure. You have particularly, of course, the membrane, and you have what we call as S protein. Okay, this is S protein because S is actually spike. You can see that the morphology looks like a spike. Okay, so this is one epitope of the COVID coronavirus. Yeah, coronavirus 2, because coronavirus is very general. And there is another epitope, okay, called N protein. Okay, so as you can see, the N protein is a bit inside the structure of the virus. And there is also, so this is another epitope. I hope that you remember when I say epitope is the one that can interact with the specific antibody. Okay, and this is also another epitope, E protein. Okay, so each of these epitope will interact with different antibody. So if you have like antibody 1, probably interacting with S protein, antibody 2 will interact with N protein, and probably if we wish to focus on epitope E, then we need another type of antibody. Okay, so you wonder how does your strip works? Okay, because this is the easiest to explain to you, right? Okay, where is so this is the basic structure for test strip. So the test strip that you use is based on immunosensor. Okay, so this is the analyte. Analyte means the sample is either you use saliva commonly. If once the test strip is quite common, everybody start to use saliva. And before that, we use throat sample, right? And then we use nasopharyngeal sample, nose, which is very uh, you know, difficult to get. I can't. uh, so uh, so what is inside this saliva and throat or nose samples? Okay, so what you actually try to get from the saliva, throat, and nose sample is, of course, the whole virus. This is the whole virus. Okay, right? But the one that interact with the epitope is here. You can see the Y structure of the antibody. Okay, so as I mentioned just now, there are three epitopes that you can use. One is S protein. The other one is N protein. And you have E protein. So if you wish to develop your test strips, you have to choose, or you can have different types of antibody to capture different types of epitopes on the antigen. Okay, so the virus is the antigen here, and these are the epitopes for that specific antibody. Now, I think you have, we are not to say immune. We heard about, okay, there's another strain of COVID virus. There's a different strain of COVID virus. There's a new deadly strain or whatsoever. What happened is, okay, when we, most of this test strip, most of the test strip that you buy and you use is based on S protein epitope. Okay, so meaning that the antibody that interact with the virus is checking on the S protein. Okay, uh, they don't check on the N protein or E protein. But unfortunately, S protein, even though it's quite common because it is outside, the structure is spike-like, and then it is outside the virus, it is easily accessible by the antibody. It is prone to, uh, that's why you have different kind of variants. Because the S protein is prone to changes in mutation. Yeah, so the protein is similar to DNA. It has nucleic acid sequences. So S protein is very prone to mutation. So when it has mutation, there is a possibility that the antibody that is developed uh, uh, at first for the S protein is no longer suitable because of the mutation. So this is the issue with immunosensor, is the mutation of the virus, especially virus, because bacteria is not that fast in mutation, but virus mutation is very common. Okay, but the other one that is good is the N protein. So N protein is stable, the epitope is stable, and it is less affected by mutation. So unfortunately, you can see the N protein is inside here. Um, Usually, uh, when you put the buffer, so it has to break down first before the N protein can be detected. That's why most of the manufacturers of test strip, they prefer to use S protein. Okay, so this is what you get in your test strip. What is on the papers? You have this area where you put the samples. Okay, and these are papers. Okay, so when paper, you have capillary effect where it moves. Depends on um, if your saliva is not that sticky, then it should be good. And you have the antibody here that responds to specific antigen or epitope on the antigen. And then when it moves, you can see that this is the one that trigger the color, which is gold nanoparticles. Okay, so when they move, you can see that you have control line and test line. Okay, so here, okay, so remember that this is the antigen. Okay, and so you have the antibody set that responds to the antigen. So there are other sets of antibody that can also respond to antigen. I mean the same antibody, but now it is placed at the test line. So when it moves, it also captures. Okay, so this antibody captures the antigen, whereas it is already captured by the antigen. Sorry, the antigen also already captured by the antibody. And you can see that this is the final structure, and this is the gold nanoparticle that triggers the color that you observe. So this is why if you have one line, single line control line, you don't have lines here, then you are negative, because this is in the control line, we have what we call as common antibody that will react, that will just react with protein here. So that's why you still have colors. Okay, so this is the concept of immunosensor. Okay, questions before I proceed? Anyone?
Prof, I want to ask for the strip. The antibodies are conjugated to gold nanoparticles. So my question is, why must we have the antibodies conjugated to the gold nanoparticles instead of the analyte go through the test line antibody directly, then it will attach to the antibody?
Oh, yeah. Okay, so the red one is the antibody, right? Sorry, the antigen, right? Okay.
Yeah.
So you can, if you imagine that there is no antibody with the gold nanoparticles, the antigen straight away will react with the antibody here. Okay. So what happened is there will be no color. Okay. So we need the antigen here to interact first with antibody conjugated with the gold nanoparticle. And this whole thing will move, and they will also interact with another set of antibody, which is specific towards that antigen. And you have this structure, final structure. If not, then you will not get color, because the one that produces the color is the gold nanoparticle. So this is what we call as tagging. So remember that the DNA biosensor, we have to put indicator, okay, to indicate the hybridization, remember? So how much hybridization happened? Is it 100%? Is it 50%? Is based on indicator. So in this case, the indicator is the color. Because without that, we cannot observe if there is any interaction or there is no interaction. Am I answering the questions?
Yes, yes, Prof, I understand now.
Uh,
Thank you.
all right, so now, so this is just a comparison, yeah? Because somebody asked, because I think if you read during the COVID, there are what we call as antibody tests or antigen tests. So what's the difference between this antibody test and antigen test? So antigen test is the one that you use to do it by the strip because you are actually detecting the virus itself. The virus is the antigen. You are infected, you have the virus inside you, you are detecting the virus. Just like the one that I have described just now. But antibody test is different. Antibody test is after sometimes you are infected, you start to produce antibody inside your body. So the antibody is now being detected. So it's a bit too late, right? It's a bit too late. Okay, if you have, anyone have dengue before? Anyone has been infected by dengue?
Yes, Prof.
Yes. How many times?
Luckily once.
Okay, please take care. If you get a second one, it's going to be a bit troublesome. Not a bit, it's going to be troublesome. Okay, for dengue, there are 4 serotypes, means that they have 4 variants. Okay, because I have twice infected by dengue. The second one was terrible. It was terrible. Okay, what happened is, if you go to the clinic, I'm not sure whether you go to the clinic testing or you just test your, what is the blood count, something, platelet, yeah? Okay, because when you are infected with virus, your platelet count is very important to check whether how bad your, your condition is. But if you go to the clinic, you can also have what we call as NS1 protein test. This is for dengue, yeah? I'm not talking about, okay. And you also can test IgG, IgG, IgM, okay? So this is the difference, okay? So NS1 protein is what we call as antigen test. Why? Because if you infected by dengue virus, okay, at the beginning, your body will start to have this protein, NS1 protein. So if you are at the early stage of dengue, so this is the right test because the NS1 protein level will go down after 2 or 3 days. Okay. So if you at a late stage of infection, this is what we call as antibody test. Because your body now is fighting. You start to produce antibody to fight the dengue virus. So now your level of antibody is climbing up, and the right test for you is antibody test. So if you test for both and you get positive for this, means that you are at very early stage. And if you get positive for this, means that you are probably a week of infection. Yeah. So that is the difference between antibody test, which is the one that produced by the body, and antigen test means that detecting the protein at the early stage of the viral infection. Okay, so of course, we want early detection because in order to know what kind of infection that we have, because viral infection can be similar in terms of symptoms, yeah, will be viral fever, but you don't know whether you have dengue, chikungunya, or this kencing tikus. I forgot what's the okay. So they are all similar. You start with fever, so you don't know. You have to do all kind of tests because if it is dengue, there's no treatment. You only treat the symptom. But if it is other disease, then there are specific antibiotic or specific drug to be given. So that's why you need to know what actually do you get. Okay. So I hope by now that you understand the principle of immunosensor. And antibody test also is considered immunosensor because it involves antibody, but there is a difference between antibody test and antigen test. Okay, so now you can see this is the comparison between the antibody test and the antigen test. As you can see, the sample for antibody test usually is blood because that is where the antibody exists in our body. It is blood. And I want to show the difference between PCR tests. Everybody talks about PCR strip test. PCR is more expensive. Why? Because they are using machines. It's not like you have the test strip only. So PCR is based on DNA. Molecular means that they measure the matching hybridization, but they are using machine, specific machine. So biosensor is the one that trying to give alternative instead of you need to use bulky machine. For example, PCR, you can have portable device to buy for you can use at homes, and it's easy, user friendly. So that's the purpose of this development of biosensor. So you can see now there is a difference between immunosensor and DNA biosensor. Okay, so of course, in terms of techniques, you have electrochemical techniques like here, okay, to be used for immunosensor. You can also use, this is considered optical because it is color based. And you can also use, instead of a strip, we can also use 96 well. So you can imagine how many samples that we can test at one time. And this is also color based, but using 96 well, we call this. Okay, so this is another types of optical, but naked eye. So instead of using test strip, you can use this 96 well. Okay. Okay, so we look at just a simple, not to say simple, on the surface of electrochemical technique, how it functions, because I haven't taught you electrochemical or optical. So we just look through what happened. So remember about the term analytes, receptors, yeah, transducers, and display. Yeah, those are the common general thing in sensor development. So you have to remember that before I can introduce you. So here the transducer is electrochemical. Yeah. And the receptor is antibody. Okay. For DNA biosensor, it is DNA. So in this case, the specific bioreceptor is the antibody. Okay. The sample can be sputum, can be saliva, can be other types of bodily liquids. blood. So those are samples. Okay, and we have now the most important is transducer and receptor. So I hope that you can recall the pictures of those basic structure of sensor and relate to this picture. So what happened here is, so this is the surface of the sensor. In this case, we are using electrode because it is electrochemistry. So we need to use electrode. And then of course, which we haven't learned is some chemistry to attach the receptor, the bioreceptor. So we need to do some chemical modification on the surface of the electrode so that we can put the receptor on the surface of the electrode. So you can see that antibody, the Y shape, yeah? Okay. So we must make sure why we need this the specific modification, because we must place the antibody correctly. Because we know that the antigen will come here. The antigen, the bacteria, the virus, because the specific interaction has to happen here. Okay. So if you get the structure, the orientation wrong, for example, this is the surface of the electrode, and you get this Y upside down, then you will not get an interaction. So you will, of course, will not get a reading. So that's why we are going to discuss this later, is how to chemically modify the surface so that we can get a correct orientation of the bioreceptor. Same goes to DNA. Okay, we haven't discussed the orientation of DNA, but now you should understand that the orientation of the antibody should be correct. It should have a correct orientation before we can develop further process. Okay, so now, so this is the antigen. In this case, because I work a lot in tuberculosis, so that's why my examples are on tuberculosis. So now you have the antigen, which is in the form of bacteria, Mycobacterium tuberculosis. And you can see now they start to interact with the antibody. It is called capture. In this case, just to differentiate, because the one that is on top of the sensor, it is called capture antibody. Okay, so the next process is similar to the strip. Okay, you have like in the strip you have gold nanoparticle. In the DNA biosensor, you have indicator, hybridization indicator. So this is the indicator. Okay, because you need that, because indicator function as the one that translate the interaction into signal. Okay, they interact. I mean, up here, up to here, of course, the interaction happened, but we cannot measure. There's no indicating that there is an interaction. So we need another system that will, you can see that this is another set of antibody called primary antibody with indicator on top of the antibody. And they also interact with the antigen. Okay. So with this indicator, we can finally have a signal generated. Okay. If we don't have this indicator, then of course, we cannot measure whether there is something happened or there is nothing happened. Okay. So that's why it is very important to understand the concept of transducer. And then we need an indicator to make sure that we can see the effect of the interaction. Okay, so this is electrical base where you have current changes, but of course we can also have the test strip, which is color based. Okay, you can see that this is the test line, this is the control line. Okay, so working with sputum is quite difficult because sputum is very sticky. So those kind of things that will affect the development of sensor. And there is also based on color generation. This is quite a difficult concept to explain because it involves enzymes. And there are, you can see that 3 sets of antibody here. So these are design, means that you want to have the best signal generation. Then those who are doing research, they design how to make it. So sometimes you use different types of antibody. So this one is more chemistry. So I'm not trying to discuss with you, but this is also another sets of immunosensor. Okay. And this is actually, for those who are familiar, it is based on ELISA. So this is what we call as ELISA plates. ELISA plates, which usually 96 well. So you can do a lot of, analysis on the same plates. So this one, these are immunosensor. So I hope that by now you understand. You also have watched the video on immunosensor. And also in the Putra Blast, I think I have given you an interactive video, not interactive, sorry, animated video on immunosensor. So next, what I'm going to discuss from 8 to 9 is aptamer. So I hope that at least between 7 to 8, you can at least watch some video on aptamer-based biosensor. You just Google them. And also in the Putra Blast, there is also one video on aptamer, which is animated video. But I suppose that you can also Google another, video from other resources so that you can at least have some idea on aptamer. Okay? That speed up your understanding on the aptamer. Okay? Is it okay, everybody?
Yes, Prof.
All right. So the next will be aptamer, right? Okay, thank you. We'll take a break. All right. As I mentioned just now, that we are going to continue to the third bioreceptor, which is called aptamer. Okay. So I hope that at least you have searched or listened to some of the videos to understand what is actually aptamer. So now we started with DNA, antibody. Now we are moving to aptamer. So as mentioned here, aptamers are single-stranded, folded. Yeah, the word here, folded. Yeah. So what is oligonucleotides? They are a series of nucleic acids. So you already knew about some nucleic acids because there are a lot of other nucleic acid apart from ATCG. There are others as well. So when we talk about oligonucleotides, means that a nucleic acid series, a series of nucleic acids joined together, and they are called oligonucleotides. Okay, so probably you have 70 nucleic acids, 100 nucleic acids joined together, they are called oligonucleotides. Yeah, so you have to understand that first. A series of nucleic acid joined together, forming oligonucleotides. So these oligonucleotides, okay, when they are folded, yeah, protein, okay, protein, oligonucleotides, if they form a bulky, we call it protein, yeah. So they tend to fold. They tend to fold. So you can see here, and you can see from the pictures that I have shown, it is not straight. It is not linear, but it is folded. Yeah, you can see the folding. Yeah, they bend. So those are characteristic of oligonucleotide. They or protein. They tend to fold, right? So what's the difference? If we have DNA strand, okay, we know already the shape is helix. It's like spring, yeah. So this is DNA. Whereas for antibody, it is Y shape, okay. So aptamer is a series of nucleic acids. You might think is similar to DNA because DNA also has a series of nucleic acids, ATCG, but for aptamer it's more than that. It's more, many types of nucleic acid. Okay, but the difference is it is folded. Okay, and folded to form a specific place, a specific place for target. Okay, so now you know DNA will, if it is DNA, it will search for matching target. Also a DNA. So DNA interact with another DNA. That is matching. Whereas antibody interact with antigen, right? Specific antigen, virus, protein, bacteria. Those are interaction between antibody and antigen. But for aptamer, we can design, okay? So we can design, so the oligonucleotides, which is a series of nucleic acid, we can design the aptamer to be specific towards the one that we wish to detect. Okay, so if you are working with DNA biosensor, of course, the target will be DNA itself, DNA with DNA. Whereas for antibody, not all can be detected. I mean, you probably can detect antigen like virus, protein, bacteria, or probably other types of biomolecular, but not so much, okay? Because as I mentioned at the early stage of this subject, that antibody has to be prepared in vivo. In vivo means that you have to like induce the production of antibody in rabbits or mice, and then we harvest. Okay, so we harvest. So it's quite difficult to have a very robust antibody, because in sensing, we want to sense metal, we want to sense drugs. So there's a lot of toxicants apart from DNA or antigen that we wish to detect. So this limits the use of DNA as well as antibody or immunosensor. So aptamer is a good alternative because we can design the oligonucleotides to be specific, okay, towards a very wide variety of analytes, including toxicants like drugs, metabolites, uh, probably chemicals, okay? Those that we cannot do with DNA biosensor or antibody based or immunosensor. So that's why aptamer is special. It is can be synthesized like DNA. DNA we can synthesize because it is a series of nucleic acid. We can just attach the nucleic acid according to the sequence that we want. So aptamer is done that way. We can do the process called CELEX. So I'm not going to talk about CELEX today. CELEX is how to design, okay? How to design the series of nucleic acid or oligonucleotides to be very selective towards the analytes. Let's say we want to detect pesticides. Okay, so there is a basic chemical structure of the pesticide. So we want to design the aptamer to be very specific. You can see that in this case it's protein, but let's say it is pesticide. Okay, so there is a specific structure of the pesticide, and we can design the aptamer. You can see that the aptamer is interacting specifically with the target, let's say pesticide. Let's say you want to detect drug, specific kind of drug. So we can design the aptamer, okay, to reactively, specifically responsive to that particular drug. Okay, it's quite similar to interaction between antibody and antigen, but antibody, the way that we produce antibody is by inducing the rabbits and harvesting the antibody. But for aptamer, we can specifically, we can chemically, yeah, synthesize, yeah, in the lab. You don't need rabbit or mice to produce aptamer. So these are the comparison between aptamer and antibody. So antibody is something that you have to use animals to produce, whereas aptamer can be synthesized in the lab, but there is a process, of course. Yeah, this is the process of producing aptamer. Of course, you can actually use rabbits, mice, where the process is generated inside animals, but this one is not necessary. You can just do it, the process in the lab, yeah, to produce the aptamer. So this is not necessary. This one can be, but it is not necessary. So it's quite robust. Okay, you can see. So I don't want to discuss this process called CELEX. The principle is easy. The principle is always easy, but the reality is quite difficult because you have to do like selectivity study, whether it does bind to the target or it is not selective. There's a series of process that you have to do, but it's a good alternative to antibody as well as DNA biosensor. Okay, so you can see another issue with using antibody is because we have to induce the production in the animal. There is a possibility of different batches will have different uh, performance. Sometimes you have a not so good antibody, so that will affect the production. Okay, if you are doing production of test strips, this one is very scary. Okay, but since like aptamer is produced in the lab, and then we are talking about sort of a chemical reaction, it is more controllable compared to inducing something in animals. Okay, and it's shorter time to prepare because you're doing chemical reaction. If you're doing in vivo using rabbit or mice, you have to wait for them to produce the antibody. So that's why, yeah, sometimes it's difficult. All right. This one is relative. When we talk about cheaper, smaller, there is relatively, yeah, because you are not using animals, it is supposed to be cheaper. Okay. So this is the Celex process. As I mentioned just now, I don't want to explain the Celex process because we are going to learn about the aptamer later. So this is how it works. Well, this is the writer is the owner of this slide is Dr. Gopinath from UNIMED. That's why you see a lot of UNIMED logos in the slides. So if you look at here, you can see that the what should I call, the maroon one is a series of nucleic acid or the oligonucleotide that is folded, which accommodates the analyte. So you can see these are the analytes, the one that you wish to detect. Okay, so the shape of the aptamer is designed, okay, to accommodate a specific interaction with the analyte. So you want to detect drugs, toxicant, uh, poison, for example, or toxin. Okay, so you can design the aptamer. Okay, so the designing process is during the production of aptamer. So called selects. There are other types of designing. It can be done via computational, which I don't have the expertise. Okay, it can be done via computational. You can do the computational interaction. You don't have to do the lab work. But still, I believe Select is the best process. Okay, right now. So that's the basis. Means that the oligonucleotide is folded to accommodate a specific site, yeah, which is specific to the analyte of interest that you want to detect. Okay, all right. So the other things are similar. Okay, you are using what kind of transducer? You want to use electrochemicals, you want to use opticals, or any other transducer, because this one has been done. I mean, you have the specific interaction between analyte and receptors, then you choose the transducer. Okay, so this is the same process that we discussed for DNA, for immuno antibody. You can pair them with transducer of your interest. Okay, so these are quite complicated, so I'm not going to discuss what is label free, what is molecular beacon. Okay, this is optical actually. This is fluorescence based, but the concept, not to say that complicated, but it is not that level to be discussed. Yeah. So this is is the same strip test by using aptamer. Okay, instead of antibody, we can use aptamer. And you can see that the normal strip test that you have used is a test line, one test line, and one control line. But actually, you can have a multiple test line, okay, which responds to, in this case, different kind of influenza. Okay, different kind of influenza, because we know that we have so many H1, H5. so so you can see that all these actors with different colors responded to different antigen or different virus, because it is designed to be that way. So the process of designing the aptamer is very important. If you have a very good selective aptamer, then you can have what we call here, when we have multiple sensing, we call it multiplex. So it is good because if you can just use single sample, but you can test many disease, right? So it is now multiplex. So you don't have to use many strips. You can just use one strip, but can tell you a series of disease or what kind of disease that you have. Okay, so you don't have to test many times. Okay, so this is another aptar sensor. So I'm not going to discuss the principle here. It's quite similar. Okay, so any question before we move on to the next topic? Can you differentiate between all these three, the DNA biosensor, immunosensor, aptamer sensor? Any question? Okay. All right. So the next one, which is very important, okay, as I mentioned a few times, that is how you put, yeah. Okay, here, you have to put the aptamer on the surface of the sensor. Same goes to the antibody just now. You have to put the antibody on the surface of the sensor. Also the DNA. So there is a chemistry involved. Yeah? Immobilization strategy is how you put the bioreceptor, in this case, on the surface of the sensor. So that is very important, because if we don't put it properly, then your test will be invalid. Okay, so I will be discussing starting from DNA, because DNA is the one that tells everything. If you be able to understand how to put the DNA on the surface of the sensor, then you understand the chemistry. You can use it for aptamer. You can use it for immunosensor. So we just discussed this, yeah? It's easy. It's how to put the DNA on the sensor surface. Okay, so the quote here: never underestimate the presence of the surface. In development of biosensors, we must take into account the effect of local environment. What does it mean, local environment? Is because the sample that you use to test, they are in complex condition. Because let's say you're using sputum, saliva, blood. You can imagine the content in the blood. There's so many other things, and you expect the small biomolecule inside that goes to the receptor and interact, because there's so many interfering factors. So imagine that you went to a very compact, a very busy market or shopping complex. Probably you have experience going for a Pasar Ramadan or or shopping for Raya. Okay, so it's very busy, it's very crowded, and you got separated with your friends. Okay, let's say. So that is the environment. And now you got to find your friend because, yeah, he or she is the one with the key to the car. Okay, so imagine that you're with a lot of people and you need to find her, you need to find him, whoever. So that's the real situation. Same goes with the analytes, the virus or the bacteria or the toxicant that you want to detect. They are in that environment, and you have to make sure that they go to the sensor. They go to their bioreceptor. So of course it is a challenge. That's why the effect of the environment, how we put the bioreceptor is very important because you want to see them. I mean, the analyte wish to see them, then they can at least go to the correct bioreceptor. Same goes with you and your friends. So how do you like decide? Okay, after everybody has finished shopping, um, how do I see you, or you actually promise somebody to meet? How do I know you? You will be wearing what color? What kind of uh, clothes? Okay, those kind of things that you have to do in sensor. You have to make sure that you are the bioreceptor is recognizable by the analyte. Okay, so the environment issues, what we call as matrix effects, is very important to be considered. Yeah. So all sensors, they have surface, whether it is optical, whether it is electrochemical. So you need to put it on the surface of the sensor. So you can see that there are a few issues. If we need to attach the bioreceptors on top of the sensors, we need to design the chemistry. Okay. In order to design the chemistry, you need to know the material, whether you have what kind of surface. You have silicon, you have gold, you have uh, non-metal. Okay, what kind of material are you using? Okay, and then after you know what kind of material, then you should know what chemistry is suitable for that particular uh, material. If you're using silicon, what kind of chemistry? If you're using gold, what kind of chemistry? If you're using glass, what kind of chemistry? So you have to know the chemistry of that suits the material. And of course, solution conditions means that it is also chemistry. Okay, probe effect, I will show you. Let me, before, what does it mean by probe effect? Okay. Um, okay, I'll just scroll for you. Okay, probe effect is if you have the sensor surface here, so as I mentioned, if you are searching for your friend, if you're within a crowded condition, if your friend is tall, definitely easy for you to search for him or her. If your friend is short, then definitely it's going to be a troublesome. That's logic. So that's why the length, okay, so imagine the DNA, the DNA. So if it is very short, okay, so this is the, let's say, DNA from the virus. Okay, let's say they're supposed to meet here. And these are the crowded, so many other proteins in the blood. Yeah. So if you design that it is too short, then it is not difficult to be approached. But if you design it a bit exposed to the surface, then probably this DNA from the virus will be able to recognize the matching DNA. Okay, so that is what we call as probe effect. Okay, a right length. Okay, so it is recognizable by the matching DNA. So that is probe effect. And another issue is, okay, restriction. Restriction, okay, whether it's DNA, antibody, or aptamer, is how much is enough for you to put on top of the surface. You want to put as much as you, if you put too much, it is not good because they start to, like, in yeah, just like a crowded place, they start to tangle, they start to uh, interact with each other, which is we don't want, because we want the DNA or the antibody or the aptamer to be recognizable by the whatever you are using here, DNA or antigen, or in the case of aptamer, it is more robust. You might have drug or toxicant. So you want this bioreceptor to be easy to spot. Okay? Easy to spot. So that is the purpose of immobilization. You want the bioreceptor to be easy to spot. It is recognizable, approachable. We don't want to be crowded. It's too short. Okay, so that's why you need to think about the restriction. How much you need to optimize? How much to be put on the surface? Stable, of course. So it is related to the chemistry. Okay. How stable the chemistry that you choose, so that once you put the bioreceptors, it will stay there. It will not leach out. Okay. I talked about accessible just now. Okay, that is very important. Okay, non-specific binding. What does it mean? Is just now I talked about there's so many other protein, probably another types of bacteria, another types of viruses, that because they are there in the environment, in our microbiomes. So they are also competing. But we designed the sensor to be selective. Okay, so we need to also make sure that this environment effect will not interfere with the reading. So that is actually very challenging, okay, to really limit the non-specific binding. Because you can imagine this is more like charges, positive, negative charges. Imagine about the hydrogen bonding. Those are, it can happen. Other proteins might also interact with the antibody we're supposed to be, not supposed to be. So those are very challenging. Okay. Okay, this is the same thing. All right. So let's look what strategies that we can use. from the simplest one. Okay. Because let's say we are using carbon. So carbon is known for easy to absorb many things. So that's why we can use simple absorption. This is, I'm just explaining to you, but we actually no longer use this kind of interaction, immobilization. We can use polymer. So this is a bit of chemistry where you use biotin. I will show you this. Biotin and streptavidin or avidin. This is a bit of chemistry. We can use nanoparticle. Just a moment. We can use nanoparticle. This is also chemistry, covalent. And this is a self-assembled monomer. This is gold, specifically for gold surface. Okay. Right. Okay. I think I talked about orientation before because as you can imagine, the DNA has to be standing up. We don't want the DNA to lie down. Simple logic, because if it is standing up, then it is easy to recognize. If it is lying down, then it's difficult to be recognized. It is difficult to be accessible. Okay. Same goes with antibody. It has to be standing up because I mentioned about that the antigen is interacting with specific epitopes. So you don't want this to be on top. Okay. And the aptamer, because it's folded structure, okay, you must make sure that the folding, that's the folded area, okay, should be accessible to the analytes, because those are the one that interact with the analyte. So if you like cover the folded orientation, then it will not interact with the analyte. Okay. Right. So this is example what I have shown just now. We want the DNA to be standing up. We don't want the DNA to be lying down. Okay. So because this will be easy for the hybridization process compared to this, right? Right. Now next step is as polymers. Okay. So what we can do, let's say we want to make sure that the biomolecule or the bioreceptor to be standing up, we can have a polymer. This one is polymer. Okay, so if you study polymer, they have this microporous, right? So this is a block of polymer. They have this cage structure, right? So you can have the DNA to be standing up, and you can control the spacing of the DNA because of the microporous structure of the polymer. Okay. But you have to think about another issue is the DNA must be accessible. Okay. If the DNA is too short, it's inside the porous structure, then the target DNA will not be able to diffuse. Okay, you don't want the DNA to be too short. You want the DNA to be accessible, easily accessible, if you are using this strategy to immobilize the bioreceptor. Okay, so these are examples of polymers. Okay, we have like chitosan, polyaniline. Why? Because polyaniline, polypyrrole, polythiophene, or known as P dot, they are electroactive. So sometimes we use it in electrochemistry. Okay. Chitosan, because of the positive charge of the glycoside. So you can see that it is not good because the DNA or the bioreceptor sort of lying down. So we don't want this to happen. So if try to avoid something like this. Okay, now is chemistry. Just now, I talked about carbon. Carbon is simple adsorption. Means that it's just adsorbed on the surface of the carbon. It does not, it's a physical adsorption. It doesn't have any chemistry. Same goes just now with polymer. Is due to microporosity of the polymer. The bioreceptors is entrapped in a cage in the microporosity, in the microporous structure. It does not involve chemistry. It's physical. But now covalent attachment. So there is simple chemistry involved. So what happened is, okay. All right, so this is an example of a little bit of chemistry. So now, as I listed just now at the beginning of this mobilization chemistry, you have to know the material used. So in this case, the material is carbon. Okay, so we know that DNA chemical structure, they only have nucleic acids, right? They have phosphates, backbone, phosphates, and they have glucose structure. So a lot of carbon, nitrogen, and phosphates. Okay, and you want to put this thing on the surface of carbon. Okay. So how do you ensure? So if you just let it there, it will interact, of course, it will physically absorb, but we don't want that because it will be disoriented. So sometimes we use a linker, okay? We put linker, okay? So this is example of linker. On top of the carbon surface, we put linker. So this is an example. There's a lot of other linker. So you can see that there is a bit of chemistry which is not explained here. You have to at least master a chemistry, how to attach amine to the carbon. There is a bit of chemistry, but I think I will have it in later slides. So now you have a linker on top of the carbon. So this is the DNA, okay, that you obtain. Let's say the DNA is specifically for dengue virus. It is designed for dengue virus. So let's say, so you can see that there is also a linker. Okay. There is also a chemistry to anchor this linker to the DNA. And usually it is done by the company. We just order. Okay, we want aminated DNA. This is called amine, right? So we want aminated DNA. We want different types of linker DNA. So that can be designed. So with this carboxylate, and we also have amine at the end of the DNA. So we can get this, what we call as amide bond. So it's not a very difficult chemistry. It's actually when we have carboxyl group and then we have amine, we can get amide bond. So it's not that complicated. Yeah. So what is EDC NHS? This is like activating reagent. Yeah. that helps to make sure that we get this amide bond. If not, then the bond sort of reversible. So you might have the bonding detached. Yeah, the covalent is not covalent. Sometimes it's difficult to get covalent bond. So once we get the covalent bond, amide bond, now you are confirm the attachment of the DNA bioreceptor on top of the sensor surface. That's a lot of work. That's a lot of work, okay? You have to think about the material, you have to think about the chemistry, and then you have to also think about the chemistry of the bioreceptor, whether it's compatible or not, and then you match it, and then you get finally the ready surface for the biosensor, okay? So this is an example of the DNA, what we call as DNA probe. DNA probe means that the series of nucleic acid, the ATCG, has been designed to specifically represent or targeting specific region of the virus of the bacteria. Okay, so this is what we call as probe DNA. Okay, so you can see that this is the original DNA structure. Okay, but there is a linker, right? Okay, so this is the chemistry that we need in order for us to attach to the sensor. Okay, so when we order the DNA probe, we have to make sure what kind of linker that attach to the DNA probe. Yeah, so these are the probe. Okay, so what is complementary, mismatch, non-complementary? If you look at complementary, the sequence, yeah, and this is the probe. Okay, okay. The match is here, A, T, C, G, and it goes on, A, T, C, G. Okay, you can try to match it, okay? So this is what we call as synthetic, because when we do research, okay, we cannot straight away test with the actual virus, the actual bacteria, because it's difficult. We start with synthetic first, okay? Okay, so this is by design to be specifically for dengue virus, let's say, by design. And then in order to make sure that our sensor is good, we react with the synthetic DNA first. Okay, and then we do all kinds of optimization, make sure that the buffer is good, the temperature is good, whatsoever. And then we move to another phase, which is the real sample, the infected blood of the patient or the cultured virus. Okay, so that's another step. You can see mismatch. Okay, if you try to match it with the probe, you can see there's one mismatch. It means that you cannot find the C for G. Okay, you try to match. Okay, and then if you look at the non-complementary, okay, this is sort of representing not non-dengue virus. You're talking about non-dengue virus, supposed to not to react. Okay, so if you try to match it, it's not a match at all. It's 100% non-complementary. So this is the one that we need to have when we've developed the biosensor. Okay, so this is another chemistry. So I mentioned just now that I didn't discuss the chemistry of carbon and amine. Straight away that I move to this and then get the DNA attached to the carboxyl group. Yeah. So if you have carbon, okay, you can oxidize. Yeah, this is acid. You can oxidize the carbon so that the carbon will have carboxyl group. So it's not that difficult. you just expose, incubate the carbon inside strong acid, and then you will get this carboxyl group. Okay. So this is EDC NHS. Sometimes it is called EDAC. So you can see from here the carbon, how to attach to amine. You need to use this chemistry first. With, strong acid. Okay, so now that you have carboxyl with EDAC, you can get same amide bond. Yeah? So don't worry about this. It's not that difficult chemistry. Okay, you need to have carboxyl, and then you need EDC NHS, and then once you have amine group, you can attach using amide bond. So there's a lot of other chemistry instead of amide chemistry. There is another chemistry called click chemistry. Okay, why I mentioned click chemistry? Okay, I hope that you can try to search what is click chemistry because it clicks. This click chemistry has won the Nobel Prize in 2023. No, not 2023. I forgot. It's 20, because 2023 is quantum dots. So click chemistry, I don't remember, but this click chemistry has won Nobel Prize in chemistry. So it's good for you to get to know the click chemistry. All right. This is another one. This now is amide chemistry, where you have the interaction between carboxyl and amine. Okay. This is what we call as thiol chemistry. Sulfur. When we talk about thiol chemistry, we're talking about sulfur. Yeah. So if you look at here, you can find sulfur, thiol, and this is amine linker. Okay. So it depends what kind of chemistry that you want, but you can see that when we talk about thiol chemistry, the material is gold. Why? Because gold, Au, our, have this affinity towards sulfur, special affinity. So that's why it's easier if you have thiol, if you have gold, then you choose thiol chemistry. If you have carbon, you have to do amide chemistry. So that depends, yeah? All right, so this is another good thing about gold. If you're using gold as the sensor surface, the thiol, they attach to gold automatically, spontaneously. Yeah, so that's why it is called self-assemble. It will self-assemble on top of gold. Okay, so you don't have to think about any other reagent. You just incubate the gold sensor surface with the thiolated DNA, okay, or thiolated aptamer, for example. So it's easy. It's automatically on the surface. Okay. So this is the protocol on how to use self-assembled monolayer. So I'm not going to discuss this. Okay. All right. Now another surface is silicon. Okay. So we have learned about carbon surface, the one that is usually common, the gold surface, and then we have silicon. Why these three? Because they are biocompatible. Because you cannot sometimes some metals or some materials, they are not compatible with protein. They are not compatible with certain biomolecule. But these materials are readily biocompatible. Okay, so silicon are well known for attachment of proteins. Yeah, so this is the chemistry for silicon. So I have this, okay, this is some basics. There are other chemistry as well. All right, you start with, this is silicon. Okay. In order to get this silanol, silanol group, the alcohol group, okay, you have to, okay, this is the protocol. So you just incubate in alcohol, okay. Sorry, sodium hydroxide. Then you will get this silanol group. So the next one is to get the amine group. Okay, this one of the steps that you can do. Okay, because once you have amine group, you can choose. You can use the amide chemistry. Okay. Oops. Okay. So you can, from the protocol, okay, first a self-assembly of ethers was formed on the electrode surface of silicon with 1% ethers. Okay, so the ethers is this one. Okay, so the ethers will, and then the second step, okay, is glutaraldehyde. Okay, so the ethers will give you the amine group. Okay, ethers, the full name is aminopropyl ethoxysilane. Yeah? So amino, the word amine, yeah? This is what you get because this is what you want. First step. Now you have amine. Okay, but in order for you to have amide chemistry, you need to react with carboxyl. Okay, so that's why you put glutaraldehyde, aldehyde. These are basic chemistry. You use aldehyde here. So in the end, you have the amide chemistry. And after you have the amide chemistry, then you start attaching the bioreceptor on top of the okay. Let's look at it. So first you get amine, and the second you get the aldehyde group, and then you can get the attachment of the bioreceptor. Okay. I think that's enough. So much of chemistry. So I think that's enough for everybody. We will discuss factors. This is just small things. What are the factors that affecting the interaction between the analytes with the bioreceptor? Okay. This is something that when you do research, you have to study all this to make sure that you get a good signal. Okay, so that will be next next topic. Okay, all right, question, anybody?